Hi everyone, welcome. Today we're joined by Dr. Stephen Schmidt, and we will be discussing the microbiome. I guess one of the unknown and exciting frontiers to solve many of the problems that we are facing in modern day medicine. Stephen, welcome. We're looking forward to what you have to say.
Thank you, young man, and hello to everyone, and thank you for that introduction. Yes, okay, so we are going to be talking microbiome, and I'm not quite sure that it's going to be weekly. It's going to depend on how hard we work and how quickly we can get these things together. But we will be doing the full thing on everything that we know in a 5-part series that I sort of worked out myself. Okay, so I am sort of 3/4 way through my life if I reach the predicted age, and I'm actually sorry that I'm not 30 years old, because I strongly believe that this concept of genetic interaction between bacteria and fungi and archaea and our bodies is the future of medicine. I truly believe that. And I'm gonna try and demonstrate to you why I say that. Every practitioner, every health person, and every individual that's concerned about his health should think about his microbiome. And that statement is a fair statement. And in the next 5 or 6, 8 maybe weeks that we're going to do this, I'm going to try and explain why I say that with some cases in the end. And Anton and Stefan, Professor Colleen is going to help us to cover this whole area from the technical stuff through to the clinical applications. I'm just going to share my screen. All right. So there's a lot of information here, and I'm not going to laboriously go through every slide. I've limited the slides for tonight, so it's not going to be too long. I will make the slides available to anyone who wants to sit and do a read by himself. The first thing that we have to understand is that I want to say that is not depicted on the slides, is that the areas of the body that is inhabited by microbacteria and microorganisms is all the areas that communicates with the outside world. So you have your upper respiratory tract, your sinuses, you have your skin, you have gynecological system, and of course the gut from mouth to rectum. So it is really the communication between the outside world and your organs. And there's an interface there which I will explain. This little picture here is done on the inside. It's a graphic. It's not a very good graphic because the colon doesn't look like this. This is a small bowel with some bugs in it, but just to give the idea. All right, so let's go through some basic things. And I'm planning to do with Anton, and the others, we're planning to do this in more or less this format. All of this will make much more sense later on. So basic information, things we call diversity, structure, and function, commensals, keystone bacteria and pathogens, clinical and therapeutic applications, products, and gut mapping. All right, so the number of microbes differs from article to article. The most recent research says there's about an equal amount of microbes to human cells, but this specific article calls it 100 trillion microbes. I don't think it really matters. It's a hell of a lot. The microbiome is called the hidden organ because it functions as a full organ. When you take the genome, the human genome, our genes, and you combine that to the microbiota, which are the organisms, that is called the microbiome. So the bugs are the microbiota, and the microbiome is the gene collection. All our metabolic features is a combination of human and microbial genetic effect. And what has brought to us in the last, I don't really know how long, last, say, decade and a half, maybe 2 decades, to this point is to identify bacteria and microbes, previous technologies had to depend on culturing. And the culturing is obviously a laborious task and a difficult task in many cases. I mean, you take Helicobacter pylori, for instance, you battle your backside off to get Helicobacter cultured. So I think the figure is about 1,000 organisms have been they've been able to culture mechanisms. But now what we're doing is we're doing sequencing and genetic identification of bacteria. So just some definitions to quickly go through. Microbiota, as I said, are the actual organisms. The bioma are the genes of the organisms and the gut. Eubiosis is a good balanced microbiome. Obviously, dysbiosis is an unbalanced microbiome with symptoms. Diversity is a very important thing. That is basically your rainbow picture in color of the wide array of your microbes. Prebiotics is basically your diet and other things that you can take in that would stimulate the growth of microbes. Sporebiotics, I haven't given a definition. It's Anton van Janssen van Rensburg's baby, and he's going to tell us a lot about sporebiotics, and it's a very important thing. But basically, you can look at sporebiotics like little infants of bacteria. So it's the baby bacteria that can grow into something else. Probiotics, we all know, those are the bacteria itself. And symbiotic is a term in research. If you read an article, they talk about symbiotics. It's basically a combination of your prebiotics, which is a food, and probiotics. We simply, according to most articles, know between 5 and 10% of what is going on in the microbiome. The majority of it simply don't know. Bacteria are very well defined. The viruses and the fungi and the archaea is not that well defined. So the makeup of the gut microbiome is bacteria, fungi, viruses, and archaea. Archaea are prokaryotic cells without nuclei, previously also called bacteria, but has since been differentiated. This is important, even though it looks very boring, because if you read these things and you read all these horrible, unpronounceable names, you basically got to always look. You can always Google this very quickly. Wikipedia is very good at this, to tell you exactly where things lie. So the kingdom is the larger group, as in bacteria. Phylum is a family description. Class is not all that important for us. The genera is sub-classification and the species. And if you had to go and look at something like Lactobacillus reuteri, which is a well-known probiotic, it would classify as a bacteria in the kingdom. It would classify as a firmicute in the phylum. Then the class is Bacilli, the genera is Lactobacillus, and the species is Lactobacillus reuteri. It's important for the interpreter of a microbiome analysis to know the stuff. And I think it's important, especially the class and the species, for everyone to know when you are going to manage someone with a probiotic or a sporebiotic or a symbiotic. This is a beautiful little graphic that I stole from the internet that actually explains to us what exactly is going on here. What are we looking for? So amplicon sequencing and shotgun sequencing are two technical terms for how they get to the actual name of the bug. Amplicon sequencing can tell you about the structure, and I'll just tell a little bit more about that, and the interaction between the bacteria. Shotgun sequencing can tell you about function and interaction, and metagenomics will collect all of this together. I'll explain it to you in the next slide and make it a bit simpler. So amplicon sequencing, or just finding the structure or a differential analysis, is basically saying to us, what is there? What are the bugs? What are the names in this collection of microbes? The metabolic pathways or the metagenomics will tell you, what do these bugs do? That's a vast field that is just scratched on the surface at the moment. So there are some of these very expensive analyses which can actually tell you this bug does this and this bug does that. So that is the function of those microbiomes, microbiota. And then a very, very important thing are the keystone bacteria. And I'm going to try and explain to you with a simple analogy. Pardon me if it sounds too simplistic or condescending. So let's say that this random group of people that I got from the internet is your total microbiota. And I'd say to you that a portion of them are English-only speakers, and a portion of them are Afrikaans-only speakers, and a smaller portion of them are bilingual. Those would constitute your phyla, or the big family, just under the kingdom. And we have 4 or 5 very important phyla in our bodies. I'll go a little bit further on that. But out of this group, you can then group them into phyla A, English speakers in the microbiome. That would be the firmicutes. Phyla B would be the Bacteroidetes. And then you've got Actinobacter, and you've got Proteobacter and Fusobacterium. Now, here's a very, very important thing I'm going to show you now, and this is why I use this analogy. If you look at these 4 gentlemen, they could be classified as language teachers. They could be either English or Afrikaans or bilingual speakers. In other words, they can be from any of the phyla. But what they are is they are language teachers. They are the keystones. So this guy can all of a sudden go away, and he was the English teacher, and then you'll find fewer English people in future, if you understand what I'm saying. So what they go across phyla, and they organize the larger groups. So if you look at what you find when you do an analysis, this is actually comes from my gut, sorry for that, but this is a bacterial species. All the species that were looked for listed in percentage representation. And if you look at a report, a good report, then you will find this little rainbow bar, and this is diversity. If we are going to do gut analysis, and you want the very first thing you want to know is, how diverse is my microbiome? There is an index called the Shannon index, which gives you a number. It's either 1, 2, 3, 4, 5, 6 or more. I'm still trying to work out exactly how that works. It's a rather complicated formula. So diversity is the #1 thing for your microbiome. These are the phyla, or the big families. Of all the names you've got to learn, you basically have to know what the firmicutes, Bacteroidetes, Actinobacteria, and the Proteobacteria are. So these 2 are the largest groups. They make up the majority of the microbiome, more or less in a ratio of 2 to 1. So 90% of the microbiome will be sorted into 2 phyla, or 2 large families, and the ratio is more or less 2 to 1. They do different functions, and we'll get to that later on. We can talk about that as well. Actinobacteria contains many of the keystone bacteria, and bifidobacteria falls into this little group, the actinobacteria, which makes up about 0.2% of your gut. The proteobacteria are usually the bad ones, and fusobacteria is not important for now. Okay, here's a depiction of the concept of the keystone bacteria. And this is a large article that I read, very complicated article, but this was the picture that came from it. And if you look at this bar here, this is a healthy person. Each of these groups would constitute a bacterial group. And you can see there is a keystone bacterium. And that keystone bacterium, why it is called a keystone bacterium? Because it regulates most of the others. So it is the language teacher that can either create more of these language speakers or fewer. In the diseased person, if you compare those two, the keystone is gone, which has led to the overgrowth of the orange bar. I think I've put that clearly. I hope you get that. So where does the word come from? This is just an interesting little observation, or where it comes from that I looked at. This is a study done by a zoologist called Robert T. Payne. And what he showed was, in a tidal pool, you have many species of sea living creatures. The purple star is the major tidal pool predator. So what he did was he removed the purple star from tidal pools. Before the removal of the starfish, these were all the critters in this tidal pool. Then he removed them, and after a year, that was what was left. And after 5 years, he only had mussels, mosselbaai. So you see these tidal pools here where I live, it's just mussels all over the show. And that is basically the loss of your keystone predator in these tidal pools. And that is an extremely important part that you have to understand if you're going to understand the microbiome and if you're going to manage people with dysbiosis. So basically, the tidal pool goes from a very varied and a diverse group of sea creatures to just a gewone ou mosselbaai poeltjie met baie mossels. The keystone bacteria are listed here. I'm not going to go through them, but you'll see the bifidobacteria is very prominent there. And the definition here, the keystone taxa are highly connected taxa that individually or in a guild or a group exert a considerable influence on the microbiome structure and functioning, irrespective of their abundance across space and time. These taxa have a unique and crucial role in microbial communities, and their removal can cause a dramatic shift in microbiome structure and functioning. And I'm going to say a little thing right at the end. Remember that little statement. Their removal will cause chaos in the gut. So where did these things come from? I don't want to go through this in detail. I just want to mention one or two things. So it takes about 3 years for the microbiome to be settled well, it was said previously from birth, but it's actually, we know now it's already in pregnancy. So it's about 3 years that it's settled. An important thing that I found in an article was that, okay, let's just go through this. In pregnancy, it's placenta, meconium, amniotic fluid that feeds microbiome to the fetus. Birth is obviously the birth canal, and there's also the skin canal with a cesarean section. And this is very important. Lots of moms have been vilified and labeled that they are a cause for their kids' abnormal microbiomes. But it's been shown that between normal delivery and C-section, the delivery equalizes after 3 years and is similar to the mother's microbiome. It takes a while longer, but it gets there. An interesting thing that I read, and I've lost the article, so I cannot quote it to you now, I'm going to find it again, is that there are many people, I think this comes from the United States, where they're actually doing a vaginal microbial transplantation for C-section babies by smearing vaginal fluids onto the fetus after birth. And then breastfeeding, first 3 years, nutrition, exposure, pets, and of course all the stuff that can destroy the microbiome after that. I've already said that these are the groups where the microbiome is present. We're going to concern ourselves with the gut. Okay, so what does it do? If you can remember this picture in your mind, you're going to understand the microbiome. Here is the lumen of the colon, and this is where all this is the electron microscopy photo. All right, so here are the bugs. This green thing here is the mucus barrier, and the blue dots over there is the colon and lymphoid tissue. Right, so if you that is just a graphic explanation of it. And bugs, mucus layer, colon cells. Now, what is very important here, there are many things that the microbiome interacts with and with the body. But two massive things that happen here is that a lot of these bacteria in certain phyla and species actually regenerates this mucus layer to keep it nice and thick. overgrowth or dysbiosis of these bugs of certain bacteria are mucolytic and breaks down this mucus layer. That's very, very important. Something that I learned not long ago, which I didn't know, was especially the thermosuits produces short-chain fatty acids, and specifically butyrate. And butyrate that is produced here feeds the colon epithelial cells directly. So if you have a lack of butyrate producing bacteria, your colon cells are under stress. One of the not the biggest causes, but a very prominent cause in constipation, because you lose peristaltic activity. And then, of course, the barrier must be a good barrier, because I'm going to show you just now. The gut microbiome from birth, actually before birth, from pregnancy, has positive effects in developmental brain development and behavior, protective effects, immune stimulatory effects, and basic role in just breaking down stuff, vitamins, production of vitamin K2, utilization of B12, feeding the colon. And of course, when things go bad, then you get diseases. This is a very complex slide, but it's actually very beautifully worked out. But I just want to highlight this part here. If you can just concentrate on this and see that the one problem that we have with interpreting the gut is the fact that the brain will control the gut in a downward control, and the gut will control the organs, and especially the brain, in an upward control. This article comes from a 46-page article that deals with microbiome and neurodegenerative disease. So the way that the gut and the brain communicates is through cytokines and toxins that go through a damaged barrier, directly through the vagus nerve, sorry for the spelling, and the enteric nerves. It cruises up in the nerves, and hormonal immune cells and neurotransmitters. I've halved that complicated picture for you just to look at a few things here. So this is a healthy gut. That is just to show that this part is the lumen and that part is the wall. You will notice the bacteria in the mucus layer. You will notice a healthy thickness of the mucus layer, and you will notice that the cells are tightly junctioned, like my fingers, so the junctions are tight. On the other side of this fence lies all the good things that has to happen, or the potential bad things that has to happen, the lymphoid tissue, the hormonal control. I'm not going to go into that now. It becomes very complicated. But basically, in this gut, you have tight junctions, you have well-fed epithelial cells, and you have the immune system is under control, and you don't have any abnormal events happening. Good fences make good neighbors. If things are right there, all of these things in place, everything is hunky-dory. Now, on this right-hand picture, you have got a thinning of the mucus layer. That says an aged gut, because it refers to neurodegenerative stuff, but a sick gut would do the same. Right, so now what you have now is you have separation of the cells, and you get leakage, the so-called leaky gut. So bugs come out here, lipoproteins, polysaccharides come out, toxins come out here. Now there is an intrusion of everything that is happening down here. Now you have the potential for increased leakage of bacteria, bacterial metabolites, LPS, toxins, DNA, mucus, all the junk that the colon can leak through that can affect the immediate immune system. It can affect the colon cells itself, go into the portal system and affect the liver, go up the enteric plexuses and the vagus plexus and affect the brain. Not going to go into these specific things. Sorry, just to point that out, that is how it works. So you can add to this list, but these diseases that are listed here actually have decent research, reproducible research, to show that dysbiosis either causes or exacerbates or contributes to all these illnesses. And pretty much you all know this describes all chronic illness in a bubble. So this is how I see it. This is my opinion, might not be completely correct, but to go from a healthy person to chronic disease, you basically have genes that could be a problem, and we know that's about 5 to 10%. All the environmental things that go wrong, which you've spoken about ad nauseum, and it works through a system of inflammation, oxidation, toxification, mineral depletion, and biopathway interruption. So these poisons are causing disease by these metabolic processes. And I actually believe that the microbiome is the gateway between those two. Because if you learn and you read about how these bugs in the colon actually causes the disease, it goes through these kinds of processes. Inflammation, oxidation, toxification, mineral depletion. For instance, if you get overgrowth in your small bowel, you get bacteria that compete for vitamin B12, and you cannot absorb your own B12 that you eat. So the microbiome and dysbiosis goes through the vagus and the enteric nervous system to chronic disease, toxin release through a leaky gut, immune dysregulation, and malnutrition, or poor absorption of nutrients. That's how it gets to chronic disease. And then through the hypothalamus, pituitary, adrenal axis, which is a very, very complicated thing, but it's quite astounding to see how that actually works. So I'm almost finished with tonight. So just a few things that is fascinating from the research and is cast in stone. And this is a very landmark, a fantastic study and a landmark study that not started the ball rolling, but started people believing in the actual effects. So the top 3 little critters here, with the stop signs here, means that they are completely sterile. Their guts have been made sterile, okay? In the left-hand mouse, what they've done is they've donated the microbiota of a normal mouse into the germ-free mouse, and the phenotype of the mouse and its offspring would be normal weighted. Then they take an obese interim, stick it into the germ-free mouse, and you have an obese mouse. And the same with anorexic mice. So what is happening here is you have a perfect model of a sterile or a germ-free mouse, put in the phenotype of weight, looking at weight differences into the germ-free mouse, and you get different body makeups. And these are actually photo of the chubby and the Twiggy. For those who know what Twiggy is, Colleen, you probably do. Mice. Just almost finished here. Just want to tell you about this incredible thing, and I've asked our colleague and friend Sabine if I could do this, and in a small group I'm allowed. Okay, so what's happening here is this is a parent and 3 siblings that was done recently, stool analyses. Sibling 1 is healthy, sibling 2 is healthy, and sibling 3 is a 19-year-old autistic boy. So they did their gut analyses, and what they found was, this is the Shannon index I was talking about earlier. So the higher your Shannon index, the more diverse your bacteria. If you can remember that very first slide that I showed of a long list of bacteria, this is all the species in your, or genera at least, in your gut that is shown here. The striking thing here is that these 2 kids have really diverse microbiota or microbiome. Mom is a little less diverse, and the autistic child has got a very low Shannon index. And what happened here was, the most incredible thing is that they took the content of one of these sibs, I think it was this one, and through colonoscopies, I think she had about 3 or 4 colonoscopies, they did a fecal microbial transplantation into our autistic child, young man, 19 years old, who was completely nonverbal and non-communicative. And this young man has improved by 80–90% to being an almost unrecognizable autistic child. So it is just astounding information. And then I don't have a slide about this, but if you go back to my slide on the, if you can just remember the slide that I showed on the keystone effect, what Sabine Hazan did, gastroenterologist and trialist in California, in the States, she was wise enough and clever enough, I can go out here. She was wise enough and clever enough to collect stool specimens from colleagues and patients prior to vaccination rollout. And she then did post-vaccination stools, 3 months, 6 months, 9 months stools. This is not published data, it's also confidential. And she's done a poster for which she won a prize. And what they showed was, I think it was 30 patients, individuals, that they did the serial testing pre and post-vaccination. 100% of them lost their bifidobacteria. And bifidobacteria is an incredibly important bacterium. It is a keystone bacterium, but it's also a bacterium that is rife in the baby and is gone in the old man just before he dies. So it's almost like your aging bacteria. So what's happened with these people, in 30 out of 30 patients who got vaccinated, their bifidobacteria was destroyed. Now, if you think about my little example of the purple star predator tidal pool, you can just imagine that this man or this person is going to be a little tidal pool with only muscles left in whatever time, unless we intervene, and that is what we are going to try and do. And this is the reason why we are setting up these little programs and these little informational things on this most incredible concept of the microbiome. Thank you, Stephanus. You can take me spotlight off me now. Okay, so if there are any questions, please shoot. We're still going to do everything in detail. There are incredible things to show you and to demonstrate from research and from articles. Almost every one of these chronic diseases has a little recognizable package of bacteria that are either in abundance or depleted. So we're going to be able to sort of identify groups of bacteria in disease. And the next thing we would like to do is then, if we can do gut analysis, is to say, okay, now we can target specific problems. And just before you ask questions, if there are, I just want to tell you this little story, and I've told it a million times, so I hope I'm not boring anyone, but I had a patient from Australia who was vaccinated, and he also had severe COVID, and he had severe autonomic neuropathy and dysbiosis. In fact, Johan, I don't know if he landed up with you. I think he might have landed up with you. Didn't Brent land up with you? Johan gone.
Excuse me, is so?
Dis Brent Feinberg by jou, daai Australiese seun?
Nee.
Okay,
Yeah.
I wanted to send him, but he didn't go. Okay, so what we did was we sorted out his autonomic neuropathy with all our beautiful little things, fire diets and all our supplements. And I think actually what happened, what worked in the end, and he got everything. He got everything. He got tocotrienol, he got GG, he got the magnesiums, he got the D, he got everything. He was a yogi and a surfer before. He was actually a healthy guy. I actually think his autonomic neuropathy was fixed with nicotine patches, because he really recovered rather dramatically after we gave him nicotine patches. But what was left was, this boy was depressed, severely anxious, and suicidal. Now, you're talking about a young man who wandered the beaches of Bondi and surfed and had 100 girlfriends. He was just a happy-go-lucky, healthy boy. And he wanted to jump off every bridge that he saw. We did a stool analysis through Sabine Hazan. She called me back, set up a consultation with me to discuss his results. And if you go down, as I sort of explained the results to you now, his diversity was fine, his phyla were in good nick, his Shannon index was normal, his Firmicute bacteroidetes ratio was normal, he had no pathogens, Helicobacter was negative. There were only 2 things that were abnormal. She does a very in-depth study. Costs you about 30,000 Rand to do. There were two things wrong with him. There was unknown viral particles, and I'm going to say something that's really confidential now, but she said to me she knows those are spike protein sequences that she saw, but she can't say it. And the second thing that was wrong is there was a keystone bacterium called Bacteroides stercosus that was in abundance. And she said to me specifically, you're going to waste your time with FMTs, you're going to waste your time with probiotics, prebiotics, symbiotics, probiotics, you call it. What you need to do is you need to give this boy colostrum. Colostrum containing all kinds of nutrients and things that mother cow gives to baby, and a lot of immunoglobulins, especially immunoglobulin G. So we got colostrum for him, made from a bovine IgG to him, made from colostrum, which I sourced here in Oudtshoorn, and Sabine sourced bovine IgG for him that is made from serum in the States. This young man has been cruising around Sydney, Perth, all over Australia for a year after he got injured. Then he got hold, well, he was introduced to me. I consulted with him. His mother flew him across to South Africa, lived in the Garden Route for a while, went to Cape Town to get better. So it's probably about 6 months that this went on. 3 weeks after he started taking his colostrum and his bovine IgG, he was back on his surfboard, back on the beach, going for the girls, and he's normal. There's nothing wrong with the man. I don't know how long the effect is, but basically I said to Mom that he must just continue taking these two products for as long as he can. Anyway, he is a happy chap. He's lost his depressive anxiety, suicidal ideations, and his heart is fixed up. And 50% of what we did was basically fixed up because we understood, analyzed, and targeted the abnormalities in his gut. Questions?
Dr. Stephen, I've got a question. This
Yes.
is fascinating. So, right when I started in COVID-19, in the pandemic, where we saw massive increases in arrhythmias during exercise within the aerobic metabolism, I specifically asked the COVID-19 task team back in the day, which was Prof. Tseke, he's head of cardiology at UCT. So he replied, and he told me that back then they were looking into the vagus nerve. And he sent me a study about that. It's linked to the vagus nerve, but now it makes sense.
Yeah, it does make sense.
Sorry?
It makes absolute sense. I mean, but sorry, I interrupted you.
Yes, but now after you explain this, it's therefore linked to the gut. And am I right? So
yes.
Okay.
I want to qualify that when it comes to vagus nerve therapy. Just so, by the way, this man to the left of me on my screen, Stefanus Neyman, knows all about it. But it's a bit more complicated than that because you also get mRNA in spike in lipid nanoparticles that Stephanie Seneff showed, which actually goes through exosomes up and cruises up the vagus nerve and does the same thing. But the vagus nerve is definitely involved. There's no doubt about that. The nicotine patches in his autonomic. Well, this young man is in his early 20s. I went to Knysna to see him there and went for a walk, and as his heart rate went up, his blood pressure and his pulse rate fell. It was dramatic that he did collapse because he couldn't keep up. So he had no appropriate heart rate and blood pressure response to exercise. And I have to say that probably everything had effect because all the stuff like the diet and the sleeping and the grounding and the earthing and the breathing and the magnesium and the vitamin D and all that stuff had sort of effect. But the most dramatic effect was after the nicotine patches, certainly of his heart.
And we also tried to look at to help his vagus nerve with specific exercise. So we got glycokineticist, some of Van Grobler
Yes.
Johan. So he helped us with some vagus nerve exercises for our diabetic patients. And some of his staff, he trained them before he passed away.
Sorry, I did forget about that, Steph. That probably also played a role because he had a few sessions with her
So
on
make a
vagus nerve therapy.
better difference, but not a lot. Why not? Because the source that was irritating the vagus nerve was still there. That's the ill gut.
That's the point. That's exactly the point. Yes, Nina, sorry.
Sorry, no, I'm never sure if I'm muted or unmuted. Obviously, I'm unmuted. So the thing with I'm not sure, Johan, how how much you understand of the vagus nerve, but if it comes to sympathetic, it's all about sympathetic and parasympathetic systems. So if the vagus nerve and the neurotransmitters and the things happening in the gut aren't right, it's going to have an effect on your whole system. It either puts you in fight or you're going to rest and repair. And I think where the shots and the things come in from, I think there are so many angles, and the one has got an effect on the other. But if you go look just only at microvascular stroking, if it happens in one place in your body, it's going to happen everywhere. And if there's microvascular stroking in the gut and you get cells dying there, then you're not going to have the right microbiome to
the other thing that's very important is that the things I didn't talk about tonight, which we'll talk about in detail, and it's boring detail, it's technical stuff, but we're going to have to go through that and learn it, is that as the adrenal axis is affected, the neuroendocrine system is affected, the hormones are affected, cortisol is affected, everything is affected that controls your blood pressure and your appropriate heart rate responses. So it's a multiplicity of things. But the way that I see it, in many illnesses, not only I'm still not worked out in my mind about this thing called long COVID, although Sabina Zahn is busy persuading me that it does exist because she also sees that loss of bifidobacteria in viral disease. And in fact, she's the one that told the Americans, the larger groups of Americans, go look for the COVID in the sewages because it's in the gut. So I'm assuming now that COVID itself, whatever it is, the spike protein, whatever it is, and this vaccination can affect the diversity and the interactions in the keystone bacteria of the gut that we know. That was shown. And believe me, you know what the pathetic thing is about you, if you want to think about whether this stuff is deliberate or not, they will not accept this incredible information as a paper from them. They will not accept it.
I'm kidding.
Yeah. Fact. So what we have to do as vaccine injury practitioners, we have to assume that every person who's had a vaccination, certainly in the Pfizer injection, because that's the one that was used, you assume that person has severe dysbiosis. And quite honestly, I'm going to make a wild statement now, but I think some people are very scared, some people are unsure, and some people are bloody desperate. You know, and if you're desperate, then I say to you, contact us. We organize a decent gut analysis. At the moment, these services are mostly overseas. Local services does not meet up to standard. Not so much in the analysis, but certainly not in the interpretation of it. But I tell you what I did do. I took a patient, and I'm sorry, Erica's not here, a patient of Erica's that Steph also knows. We had one of these analyses where you get 20 pages of bacteria listed in lists and lists and lists and after. I mean, you don't know what the hell's going on there. But me studying this thing, I took her report, I analyzed it, and she had a firmasuit bacteroidetes ratio of 49:1. It has to be 2:1 if you remember the slides, if you were awake. 2:1. 60:30. She had a 49:1 firmasuit, complete overgrown of firmasuits. And here's the kicker. The firmasuit is the obesogenic, diabetogenic phylum, and she is grossly obese, and she's a very difficult diabetic to reverse, because she's on the FIRE program. We're doing everything we can to try and reverse her. And I've now identified this woman's problem.
I mean, she's one of those non-responders. It's just
She's a non-responder.
so your non-responders in diabetic reversal, your typical Ozempic candidates is actually a gut problem. So just give them stumps to correct their gut and get this ratio ready instead of injecting them with Ozempic. So now I understand why they won't accept any papers, because
yeah, they won't. I mean, and that is another thing. If you are confronted with this is fascinating stuff from all the stuff I've read in the last few weeks, is that your tendency would be, okay, go give some Bacteroidetes species to balance the Firmicute species. But with what I explained to you tonight, what you actually need to do is you need to go and target the keystones that will balance the Firmicutes. So we've got a very definite, I can't wait to see her and explain this to her and then see what the response is. I'm definitely going to collect that data and present that at some stage. I'm not saying, I'm praying it's going to work, but she has an incredibly serious dysbiotic gut analysis. And I also want to say to Helen, you and Marie, who might have requested these kinds of tests, I know Erica does quite a bit of it, and you get these like 20, 30-page stuff, and you don't know what the hell's going on. I'm quite happy, me and Steph and Anton are quite happy to sit and look at this stuff, analyze it for you guys, and come up with reasonable answers.
Right.
Because it has to be done properly. It's this sort of, here is your result with 1,000 different species listed on 20 pieces of paper is useless.
How do you analyze a telephone book? No,
exactly. The only thing you can derive from that, basically, for sure, is you know your Helicobacter status, and you know if there are pathogens that you can manage the pathogens with antibiotics. But for the rest, you can't make sense of what they tell you.
So when are you going to open your lab in South Africa, Stephen? Can we not stop getting funders for this?
You're such an astute young lady, you know.
Well, that's where we've got to go. I mean, we can't carry, I don't even do these stool tests anymore because I haven't got a clue what the hell I'm supposed to do with that damn thing. It's just too much. You haven't got a clue. If you're not as keen as on, how the hell are you going to know who's who in the zoo? Because it is a zoo when you get it.
It is a zoo. Watch the space, Ellen. It's in the making. We're there. We're almost there.
Okay, great.
If you see many autistic and spectrum kids, the parents actually, once you do their first parasite cleanse, they send you pictures of the stools.
Yeah.
No?
That's how crazy it is. But you know what, the autism thing is an incredibly fascinating thing. But it's going to take a lot of work because it's a very complicated article. Colleen, I think I sent you that article, the interaction or the relations and the causality or the correlation between the gut, Parkinson's, ALS, dementia, Alzheimer's, Lou Gehrig. It is incredible. It's absolutely incredible. I learned this stuff 2 years too late. Otherwise, I could have probably helped my sister. Both the vaccination and the dysbiosis and the relation, especially with this direct gut-brain axis and the enteric nerve plexus, and of course then the hypothalamus pituitary adrenal gland axis. You know what was so incredibly fascinating, and I'm sitting here and I'm getting a little bit emotional, was the fact that my sister had these incredible episodes of what they called epileptic fits, but they weren't epileptic fits. She just completely dropped her blood pressure into the ground, like a pow, like that. It just went away and then recovered again. And what she had was, they weren't arrhythmias. She had these alters done. It wasn't epileptic fits. It wasn't seizures. It was nothing else. It was just a complete drop of autonomic control. Dramatic stuff. And I believe her adrenal glands were shot, or at least between the pituitary and the adrenal glands in the hypothalamus. And if you go through that article, it's it's incredible. You cannot ignore a person who has early dementia signs, an Alzheimer patient in full-blown Alzheimer's or Parkinson's. You cannot ignore those people anymore because you can actually likely help them, at least if you can improve their quality of life. I need another lifetime because I reckon if I was a student now and I knew what I know now, I would everything I do, I would go and specialize and get a PhD, and I would concentrate on this thing and this thing, not only, but it's such a crucial, critical organ. And then not even talking about the mouth and the heart, the sinuses and the lungs and the heart, the gut and eczema and allergies, the vaginal stuff, I mean, the psoriasis in the skin, the eczema in the skin, all those things are well described. There's a wealth of information out there. I'm talking too much.
Dr. Stephen, I've got another question.
Yes.
So can this explain my observation for the last 5 years? Because we're trying to understand why would the arrhythmias in the atria and the ventricle be 7.5 times more in males and females, specifically in the lipid metabolism. So I asked Prof. Larry Dursteen this, and he did his PhD in exercise in the lipid metabolism in the 80s, and he is the global expert in this, and he couldn't explain. So the only thing that we could use was mitochondrial dysfunction, because it was linked to mitochondria, because suddenly you're exposing this. But now I'm starting to think it should then therefore be linked to the gut some other way. But how do you, or how does this come together?
Okay, so I am not the expert. I'm a student at the moment. I'm an in-depth student, but I can certainly go and look for you. But you know what is incredible whenever you hear of anything? I mean, this most amazing thing is this SSRI and sexual dysfunction, post-SSRI sexual dysfunction. I just take these things and say PSSD and the microbiome. There are articles written on how this works, and there's a definite gut connection. So I WhatsApped Sabine and I said, listen here, do you see, what do you think about PSSD? She said it's an absolute gut thing. She hasn't answered me afterwards as to why she says that. And the same applies to the Propecia, finasteride, you know, the hair growth stuff, Propecia, that is used and gives young men basically complete sexual numbness. Finasteride, sexual dysfunction, and SSRI, sexual dysfunction, involves exactly the same group of bacteria. Fascinating.
I have an interesting, there's an interesting woman in Stellenbosch who deals a lot with kids with all these, they are, what do they say? The one's a squirrel.
On the spectrum.
No, they identify as the strangest things. They actually believe that.
Oh.
She said, and she's very much into toxicology, first of all, and then obviously everything that goes along with that, the diet, the whatever we put into ourselves, what you put on yourself. So the whole spectrum of toxicology. But she said once you can sort out these schizus metabolics, they actually return to normal. They start identifying as themselves again. And I think like just the world has lost its plot. We should get back to basics. Start growing your own stuff. Start milking the cow in your backyard. Stop.
Yeah, you must also cancel their TikTok accounts, because I think they learn things on TikTok. All right, guys, I am at the end of my tether here today. You can stop the recording, Steph.
Yeah, okay, let's just thanks, Stephen. So appreciate what learning from you. We are all learning. Yeah, thanks so much. Until we meet again to discuss the microbiome. See you soon.
Pleasure.